Welcome back to another deep dive. Today, we're going to be exploring the world of process optimization, specifically how scientists use something called design of experiments,
Okay.
or DOE, to find the absolute best way to make things.
Right.
And I'm not just talking about baking the perfect cake here,
Yeah.
although DOE can certainly help with that. We're talking about high-stakes processes like developing life-saving medications or creating cutting-edge materials.
Yeah, absolutely.
Now, you might be thinking, experimentation. Isn't that just trial and error?
Yeah.
Well, not exactly. DOE is kind of like a supercharged version of experimentation.
Yeah.
It's about being strategic, not just randomly changing things and seeing what happens, but carefully selecting which factors to adjust and at what levels to get the most valuable information out of every experiment.
Right.
It's about being efficient. It's about saving time and resources. And ultimately, it's about making better products. And to help us navigate this complex world, we have an expert joining us today, someone who actually uses DOE in their work. So to get started, can you give us a quick overview of the different types of DOE we'll be exploring today?
Sure. Three main types we're going to focus on today are factorial designs, fractional factorial designs, and something called response surface methodology, or RSM.
Okay.
Each one has its own strengths depending on the complexity of the process you're trying to optimize.
Okay. Let's break those down one by one. I think most people have some intuitive understanding of experimentation, right? You change something, you see what happens. How's a factorial design different from that?
Sure. So let's imagine you're trying to develop a new drug, let's say dronabinol, a synthetic form of THC.
Okay.
You want to optimize the synthesis process to get the highest yield possible while ensuring the final product is pure and safe. Now, there are a lot of factors that can influence this synthesis, things like temperature, pressure, the concentration of different ingredients, the list goes on. A factorial design would involve systematically testing all possible combinations of these factors at different levels.
Okay.
Think of it like a recipe where you try out different amounts of each ingredient to see which combination gives you the tastiest result. Except in this case, the taste we're looking for is the optimal yield and purity of our drug compound.
So if we have, let's say, two factors, temperature and concentration, and we test each at two levels, that would give us 4 different combinations to experiment with.
Precisely. And a full factorial design would require us to run experiments at each of those 4 combinations. Now, this can get pretty complex if you have more factors or more levels to test.
Sure.
And that's where fractional factorial designs come in.
So it's like a shortcut.
Yeah,
Yeah.
it is. But it's a smart shortcut. It lets you test a carefully selected subset of those combinations while still giving you enough information to draw meaningful conclusions.
Like strategically sampling the cookie dough instead of baking every single possible batch.
Exactly. And then we have response surface methodology, or RSM, which takes things to a whole other level.
Okay.
This is where we really start to fine-tune things and find that sweet spot where everything works perfectly.
Okay. I'm definitely intrigued by RSM. What makes it so special?
Well, imagine you're trying to climb a mountain.
Okay.
You don't just randomly start walking in any direction, do you? You look at the terrain, you assess the slopes, and you try to find the path that will lead you to the highest peak. RSM works in a similar way. It allows us to create a map of the response surface, how the output of our process changes as we adjust the inputs. And once we have that map, we can use it to navigate our way to the optimal settings.
So in the case of our dronabinol example, we could use RSM to find the perfect combination of temperature, pressure, and concentration that gives us the highest yield of pure dronabinol.
Precisely.
Kind of like pinpointing the summit of our mountain.
Exactly. And the beauty of RSM is that it allows us to optimize for multiple responses simultaneously. For instance, we might also want to minimize the formation of any harmful byproducts during the synthesis.
So we're not just looking for the highest peak, but the peak with the most breathtaking view.
Exactly. RSM helps us find the settings that achieve the best balance across all our desired outcomes.
Now, I've heard this term quality by design, or QBD, thrown around a lot in the context of DOE.
Yeah.
Can you tell us how those two concepts are connected?
Sure. QBD is all about building quality into a product from the ground up.
Right.
It's about understanding your process inside and out, so you can control it effectively and consistently produce high-quality results. And DOE plays a key role in achieving this.
Okay.
Think of DOE as the toolbox that allows you to collect the data you need to implement QBD principles. It helps you identify those critical process parameters, the things that really matter for product quality. and then figure out how to control them to ensure consistent, high-quality results.
So it's like having a blueprint for quality.
Exactly. DOE gives you the insights to build a robust and reliable process, one that consistently delivers the quality you need.
Okay, that makes a lot of sense.
Mm,
So we've got our different types of DOE, our tools for exploration and optimization. We've got QBD, our guiding principle for building in quality.
right.
It sounds like a pretty powerful combination.
Yes.
But before we dive into, specific examples, let's take a step back and talk about the bigger picture.
Right.
Why is DOE so important in the first place? I mean, haven't people been experimenting and making things for centuries?
Of course they have. But the world is becoming increasingly complex. The products we're developing are more sophisticated.
Right.
The processes are more intricate. The stakes are often much higher.
Yeah.
We can't afford to rely on trial and error anymore, especially when we're talking about things like pharmaceuticals or advanced materials.
So DOE is like a necessary upgrade for the modern world.
It is. It's about being smart and efficient with our experimentation, maximizing the knowledge we gain from each experiment and minimizing the resources we use. It's about making informed decisions based on data, not guesses.
Right.
And ultimately, it's about creating better, safer, and more reliable products for everyone.
I love that. Better, safer, and more reliable. So are you ready to roll up our sleeves and dig into some real-world examples? I'm particularly curious about how this all plays out in the development of things like synthetic cannabinoids and psychedelics.
Sure.
Those seem like areas where precise control and optimization would be absolutely essential.
Absolutely. Let's explore how DOE can be used to optimize the synthesis of compounds like dronabinol or psilocybin, the active compound in magic mushrooms. These are areas where even small variations in the process can have a significant impact on the final product.
Right.
And DOE gives us the tools to navigate that complexity.
Sounds good. And for our listeners, get ready to enter the fascinating world of controlled experimentation. We'll be right back after a quick break.
All right, let's jump back into this world of DOE and see how it can be applied to the synthesis of compounds like dronabinol and psilocybin.
Yes, please. These are fascinating areas, and I'm eager to learn how scientists use DOE to optimize these processes.
Well, just like with any chemical synthesis, there are a multitude of factors that can affect the final product. Temperature, pressure, reaction time, the concentration of reactants, even the types of solvents used can all play a crucial role.
It sounds incredibly complex, almost like trying to juggle a dozen balls at once.
It can be, yeah, but that's where the power of DOE comes in. It helps us to break down this complexity and systematically explore how each of these factors and their interactions influence the outcome of the synthesis.
Okay, so let's say we're trying to synthesize psilocin, the active compound in magic mushrooms. Where do we even begin?
Well, we'd start by identifying the key factors that we believe might have the biggest impact on the yield and purity of our psilocin. This might involve reviewing existing scientific literature, consulting with experts, or even conducting some preliminary experiments.
So it's like doing some detective work before we even start the main experiment.
Exactly. Once we have a good understanding of the potential factors involved, we can start designing our experiment. Let's say we've narrowed it down to two key factors: temperature and the concentration of a specific catalyst.
Okay, and I remember you mentioning that each factor is tested at different levels. How do we decide what those levels should be?
Well, that's where our initial research and expertise come into play. We need to choose a range for each factor that's both practical and likely to reveal meaningful information.
So for temperature, we might choose a range that aligns with what's feasible in our lab setup. And for the catalyst concentration, we might look at what other researchers have used in similar syntheses.
Exactly. Let's say for temperature, we choose 60 degrees Celsius and 80 degrees Celsius as our two levels. And for the catalyst concentration, we select 1% and 3%. This gives us a total of 4 different combinations to test.
Now, if we were to run a full factorial experiment, we'd test each of those 4 combinations. But I remember you mentioning fractional factorial designs as a way to be more efficient.
Right, exactly. If we had more factors or more levels to test, a full factorial design could quickly become overwhelming in terms of the number of experiments required.
Sure.
Fractional factorial designs allow us to strategically select a subset of those combinations, reducing the experimental workload without sacrificing too much information.
So it's about finding that sweet spot between effort and insight.
Absolutely. And the choice between a full factorial and a fractional factorial design depends on a few factors like the complexity of the process, the resources available, and the specific questions we're trying to answer.
Now, you also mentioned RSM as a more advanced technique for optimization. How does that fit into the picture?
RSM is particularly useful when we want to fine-tune our process and find the absolute best settings. Remember that mountain climbing analogy?
Yes, I do. We're looking for the highest peak on our response surface.
Exactly. RSM helps us map out that surface in more detail. Moving beyond those discrete points, tested in factorial designs to explore the entire landscape and identify the optimal settings.
So it's like having a more detailed topographic map, allowing us to navigate the terrain with more precision.
That's a great way to put it. RSM often involves building a mathematical model based on the results of our initial experiments. This model allows us to predict the outcome of the synthesis at different settings and to identify the region where those optimal settings are likely to be found.
So we're essentially using data to guide our search for the perfect recipe.
Precisely. And one of the key advantages of RSM is that it allows us to optimize for multiple responses simultaneously.
So in the case of psilocin synthesis, we could potentially maximize the yield while minimizing the formation of any unwanted byproducts.
Exactly. We could even factor in other responses, such as the cost of the process or the environmental impact, and use RSM to find the settings that best balance all of these considerations.
This is starting to sound incredibly powerful, almost like having a crystal ball that shows you the best possible outcome.
It's not quite magic, but it's certainly a powerful tool. And when combined with the principles of QBD, DOE becomes even more effective in ensuring the quality and consistency of the final product.
Right, QBD was all about building quality into the process from the very beginning.
Exactly. And DOE provides the data and insights we need to implement QBD effectively.
So could you give us an example of how this might play out in the real world? Let's say we're developing a new dronabinol-based medication for the treatment of chronic pain. How would DOE and QBD work together to ensure the quality of that medication?
Well, in that scenario, we would start by identifying the critical quality attributes, sometimes called CQAs of our medication. These are the characteristics that are essential for its safety and efficacy, things like the purity of the dronabinol, the stability of the formulation, and the consistency of the dosage.
So these are the non-negotiables, the things that absolutely have to be right.
Exactly. Then, using DOE, we would systematically explore how different factors in the manufacturing process influence those CQAs.
Okay.
This might involve looking at the impact of temperature, pressure, mixing speed, or even the type of equipment used.
So we're essentially creating a map that shows us how tweaking those knobs and dials in the process affects the final product.
That's right. And once we have that map, we can use it to define a design space, a range of settings for each factor that will consistently produce a high-quality product that meets all those critical quality attributes.
So it's like drawing a safe zone on our map, a region where we know we'll be producing good results.
Precisely. And this design space is a crucial element of QBD. It provides a clear roadmap for manufacturing, ensuring that the process remains within those safe and effective operating ranges.
This all makes a lot of sense, but I'm wondering, how do we actually know that our DOE experiments are giving us reliable results? I mean, there's always the possibility of error or variability in any experiment.
That's a great question, and it's something that scientists take very seriously. DOE methodologies are designed to minimize error and variability.
So how do they do that?
Well, for one thing, DOE experiments often involve replicating each experimental run multiple times. This helps to account for random variations and provides more confidence in the results.
So it's like taking multiple measurements to make sure we're getting an accurate reading.
Exactly. And there are also statistical techniques that are used to analyze the data and assess the significance of the results.
So it's not just about collecting data, but also about interpreting it correctly and making sure that our conclusions are actually valid.
That's right. DOE is a rigorous and systematic approach, and when done properly, it can provide a very high level of confidence in the findings.
Okay, I'm starting to see how all these pieces fit together. We've got our DOE tools for exploration and optimization. We've got QBD guiding us toward quality. And we've got statistical methods to ensure the reliability of our results. But before we wrap up, I'd love to hear a bit more about how DOE helps us not only optimize a process, but also identify and manage potential risks. After all, when we're dealing with things like pharmaceuticals, safety is paramount.
Absolutely. That's a crucial aspect of DOE and QBD.
So let's dive into that in part 3. We'll be right back after a quick break. Welcome back. We're diving deeper into the world of DOE and how it's not just about optimizing processes, but also about ensuring safety and managing risks. So let's pick up where we left off. We were talking about the importance of risk assessment in pharmaceutical development. Can you elaborate on how DOE helps us identify and mitigate those risks?
Sure. When developing a new medication, it's crucial to identify any potential hazards associated with the manufacturing process.
Okay.
This could include things like the formation of harmful byproducts, degradation of the active ingredient, or even contamination during production.
So it's about looking for those potential weak points in the process, the things that could go wrong and impact the safety or the efficacy of the medication.
Exactly. And DOE provides a structured way to explore those potential risks. So by systematically varying the process parameters, we can observe how those changes affect not only the desired product, but also the potential formation of impurities or degradation products.
So it's like running a stress test on the process, pushing it to its limits to see where those vulnerabilities might lie.
That's a great analogy. And by identifying those risks early on, we can take steps to mitigate them. This might involve adjusting the process parameters, implementing additional purification steps, or even redesigning the manufacturing equipment.
So it's about being proactive, not reactive.
Precisely. DOE, when combined with risk assessment, allows us to build safety into the process from the ground up. And this is particularly important when we're dealing with compounds like synthetic cannabinoids or psychedelics, where the potential risks and side effects are often not fully understood.
Right. That makes a lot of sense. So can you give us a specific example of how this might play out in practice? Let's say we're developing a new dronabinol-based medication for the treatment of chronic pain.
Sure. In that scenario, one of the potential risks we'd want to explore is the degradation of the dronabinol molecule during storage.
Yeah.
Over time, dronabinol can break down into other compounds, some of which might be inactive or even harmful.
So we need to make sure that the medication remains stable and potent over its intended shelf life.
Exactly. We could use DOE to investigate how factors like temperature, light exposure, and the packaging materials affect the stability of the dronabinol. By systematically varying these factors, we can determine which conditions accelerate degradation and which ones help to preserve the integrity of the molecule.
And that knowledge would then allow us to design a storage and packaging strategy that minimizes degradation and ensures the medication remains safe and effective for as long as possible.
That's right. We might find that storing the medication at lower temperatures or protecting it from light significantly slows down degradation. We could then use this information to develop appropriate labeling instructions and packaging to maintain the quality of the product.
So it's about using DOE to not only optimize the manufacturing process, but also to optimize the entire life cycle of the product, from production to storage to patient use.
Exactly. It's a holistic approach to quality and safety. And this approach is not limited to pharmaceuticals. DOE can be used to assess and manage risks in any complex process, from manufacturing to food production to environmental remediation.
So if we were developing a new manufacturing process for a high-performance material, we could use DOE to explore potential risks, like worker exposure to hazardous chemicals or the environmental impact of waste disposal.
Precisely. DOE gives us a framework for thinking systematically about risks, identifying potential hazards, and developing strategies to mitigate them.
It's like having a risk management tool built right into the experimentation process.
That's a great way to think about it. And by proactively addressing risks, we can ensure that our processes are not only efficient and effective, but also safe and sustainable.
This has been a truly enlightening deep dive into the world of DOE. It's amazing to see how such a seemingly technical approach can have such a profound impact on the quality and safety of the products we use every day. So for our listeners who might be feeling a bit overwhelmed by all this information, what's the one key takeaway you want them to remember about DOE?
I'd say the most important thing to remember is that DOE is not just about finding the best settings for a process, it's about understanding the process itself, how different factors interact, how those interactions influence the final product, and how we can control those factors to achieve our desired outcomes.
It's about being intentional, systematic, and data-driven in our approach to process development.
Exactly. And whether you're a scientist developing a new medication, an engineer designing a new product, or even just someone trying to perfect your baking skills, the principles of DOE can be applied to make your work more efficient, effective, and ultimately more successful.
Well said. I think our listeners are now equipped to go out and start experimenting with a newfound appreciation for the power of DOE. Thank you so much for joining us on this fascinating journey.
It was my pleasure.
And to our listeners, keep exploring, keep learning, and keep diving deep. We'll see you next time on The Deep Dive.